Well, good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Establishing New Standards in Efficient Single-Cell Cloning of hiPSCs. My name is Samaya, and I'll be your X-Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved, so please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require any assistance, you can contact me by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please also note that this event will be recorded and made available for future streaming on xtalks.com. At this point, I'd like to thank Solentim, who developed the content for this presentation. Solentim is a trusted global leader for solutions to create, isolate, and characterize the highest value cells for the development of new biological medicines, including cell and gene therapies, monoclonal antibodies, vaccines, and stem cells. For their customers, which include most of the world's leading biotherapeutics companies, they are the partner of choice for facilitating the highest level of assurance in cell line development for therapeutic protein and viral vector production. Their portfolio of proven and innovative technologies, combined with their unparalleled experience, ensures that customers achieve accelerated master cell bank development and are confidently prepared for regulatory review. Now it's my pleasure to introduce our speakers for today's event, and our first speaker is Phil Manos. Philip is a stem cell expert with a successful history of technology development and business operations within both academic and commercial organizations. Since entering the human-induced pluripotent stem cell field in its infancy in 2008, he has developed a strong publication record focused on hiPSC technology, including reprogramming via modified mRNA for derivation of the first clinically relevant hiPSCs. Philip began his career at the Harvard Stem Cell Institute HESC core facility. He then went on to help establish reprogramming and hiPSC disease modeling platforms at both the Novartis Institute for Biomedical Research and the Neural Stem Cell Institute before serving as the Director of Operations for Stem Cultures. Our second speaker is Ian Taylor. Dr. Taylor has a PhD in biochemistry. He has more than 25 years working in the conceptualization and commercialization of novel high-value life science instrumentation. Prior to joining Solentim, when it was founded 10 years ago, Dr. Taylor was commercial director of Genetics PLC, where he was responsible for the development and sales of the ClonePix FL for selection and isolation of high-value monoclonal antibody-producing clones. Prior to that, Dr. Taylor was part of the senior leadership team for PerkinElmer Life Sciences. So now, without further ado, I'll pass control to our first speaker, and that's Ian. So Ian, whenever you're ready, you can go ahead and get started.
Right. Thank you very much for the introduction. I'm going to just set out what the agenda for today is, first of all. So I'm going to briefly introduce the technologies, the platform that we're going to talk about, and then I shall hand over to our main speaker of the day, Phil Manos, who will talk about his work he's done using the VIPS instrumentation with a range of human-induced pluripotent stem cells. Phil will sort of set out some of the general challenges in the field for working with iPSCs. He'll then take us through the sort of optimization steps that he developed working with a cell line to optimize the VIPS and take it through the whole process. Then he did a broader validation study he'll talk to, excuse me, with a number of stem cell lines. And then Phil will sum up that data and talk about how this new workflow can impact different types of workflow using stem cells, whether that's gene editing or reprogramming, etc. We'll then switch back to me for the final part of the talk today, where I'll really set out Solentim's strategy of how we're going to support regulatory and clinical aspects with an eye on those of you that are working on and developing stem cell therapeutics and will require that assurance and support. And then finally, we'll just touch on some next steps for people who are interested to follow up after today, how you can get your hands on the technology and try it out. So let me start with the introduction to the technologies. So the first component of this is actually the instrumentation. We see a picture of it there on the bench. It's a benchtop instrument. VIPS stands for Verified In Situ Plate Seeding, and it's a two-in-one system. It does both the single cell seeding, but also importantly from a regulatory perspective, confirmation in the whole well using whole well imaging of the arrival of the cell in the well. Whilst stem cells is a new sort of workflow for us, I want to give the audience reassurance that the VIPS product itself has actually been a commercial product for over 3 years now, and has been successfully used by many of our biotherapeutics companies for single cell cloning of therapeutic CHO cells and HEK cells for gene therapy. So it's tried and trusted. The key elements of this system are it's a very high seeding efficiency system, giving in a range of 70 to 90% seeding efficiency. But importantly, it's a very gentle system, and that really plays into one of the important aspects of this workflow in stem cells, that it ensures high viability and outgrowth into colonies. The system also, I've already touched on how we're going to address or work with stem cell therapeutics. The system is also geared up for full documentation of clonality, and that's going to be important when putting together a package for the regulator for an IND package, for instance. The system, as I say, is benchtop. It's about the size of a microwave, a small microwave, and it would go typically in a Class 2 container or biosafety cabinet. It currently supports 96 well plate format, but 384 has literally just been launched as well, so you have the choice of both formats. And really, in summary of this slide, we're providing what we call a double lock of clonality assurance, that you get proof of the single cell in a single cell droplet, but also the whole well image after media addition. And I'll show that in the following slides. So just finishing off with some other comments on the VIPS. It's as I said, it's a low pressure gentle system delivering typically a 20 nanoliter droplet. It then uses the images from beneath the microtiter plate, and it does what we call Z-stack or Z-stack imaging. So it takes slices through the droplet and then uses clever imaging software to confirm the presence of a single iPSC in that droplet, and to resolve it from other facets, for instance, bubbles. It does automated image analysis for a single cell detection of iPSCs. It does the in-well assurance record at what we call day 0, and then does daily whole well imaging as well, so tracking that single cell growing into 2 cells, into a cluster, into a colony. And as I said, it compiles that clonality report. So one of the key things we've done as we've moved from CHO cells is we've actually trained the VIPS with libraries of iPSC cells so that it can specifically recognize these types of cells in the droplet and use that intelligence. So this is now the workflow using the VIPS. So what we're doing first is, and if I can just maybe get a pointer. Yeah, so there we go. Just up here at the top with the laser pointer, we actually dispense the cell droplets into a dry well, and we do a sort of array format whereby we start off, for instance, in this position here, and we put down a 20 nanoliter droplet, and then we immediately image that droplet using the Z-stack imaging to see if there's a cell in it or not. And that's a droplet image you're seeing below. If we've got detection of a cell, there it is in the droplet. If there's no cell in that first droplet, apply a second droplet on top, and then we can continue out of third and fourth droplet into that loci to see if we've got a cell or not. If you've got the concentrations pretty much spot on in the cell reservoir, you would typically get a cell identified in the first 3 to 5 droplets on that particular spot. The other imaginations, if you had your cell concentration a little bit out of kilter, you could actually go up to 4 positions with 4 spots in each loci, so 16 droplets, but it wouldn't make it a particularly efficient process if you did it that way. And then importantly, we process a single well at a time. So as soon as we've identified the cell, and it's identified here, we fill it up from a separate nozzle with matrix and media, and that's where our MatriClone growth media will be coming from. Right, let's go on to the next slide. So just to finish off on that, I really want to focus on the right. This is sort of showing what's happening in real time as you're seeding that plate. So just to explain the difference in this slide, a green spot is where we've got a single cell confirmed in the well immediately as we're processing the plate, and a red well with a white cross is where we've got more than one cell, so it could be a doublet or something like that. But this whole processing of this plate would take roughly 10 minutes, and depending upon the cell type, we'd get between a seeding efficiency of between 70 to 90%. The key thing with this is we mentioned efficiency, but it's also the droplet image that you see over here in the middle, because the droplet is the kind of key additional image here, and it's the first image in that clonality report for the regulator. And I always call it the inception of clonality. That's the first time you see the single cell in the well, and it's in that droplet. So at this point, I've talked about the instrumentation. I just want to introduce the MatriClone. This is a brand new matrix that we're offering. It's very much, I think of it as a combination of the VIPS with the MatriClone is the power of the success in this workflow. So it's a specific matrix that we've launched at Solentim for both the seeding, so it comes in through that separate nozzle at the point of processing the well in solution, and it helps the growth and viability of the iPSCs. Importantly for people working in the clinical area as well, it's defined, it's animal component free, and it's based on a recombinant protein. And importantly, in the way that Phil's used it in his work, is we've developed it for use in a soluble form, which has a number of advantages. It means you save hours of preparative work before you do the experiments, where you no longer need to pre-coat plates. But by using it in solution, you can also use it at much lower concentrations as well. As we saw, it's dispensed automatically through the VIPS from a separate channel on the instrument. It supports a high viability, and we've previously shown, I think we spoke at iPSCs in June, and you get typically 4 to 5 more colonies per plate using the VIPS with the MatriClone compared to limiting dilutions. And importantly, for cell metric customers that don't have a VIPS yet, you can get your hands on MatriClone straight away and start using it in either a soluble form or as a plate coating. So that's the end of my introduction. So I'm going to hand back to the organizer now, who will do poll question number 1.
Thank you, Ian. So that does bring us to our first poll question. Audience members, you should be seeing that poll question on your screens. So that question is, what matrix do you currently use for your iPSCs? And your options are MatriGel, vitronectin, laminin-based, or other. So go right ahead and click on the option that suits you best. And once again, that question is, what matrix do you currently use for your iPSCs? And your options are MatriGel, vitronectin, laminin-based, or other. So I'll give you a couple more seconds to answer that, and then we'll go ahead and share the results. Okay, fantastic. It seems like the majority of audience members have voted, so I'll go ahead and close the poll and share the results. And the results are as follows. So vitronectin was the majority of votes went to. That was at 33%, then followed by MatriGel at 31%, laminin-based at 19%, and then other at 17%. Thank you for participating, audience members. Now we'll send controls to Phil. So Phil, whenever you're ready, you can go ahead and get started.
Great, thank you very much. Hello, everybody. First, I'd like to thank Ian and this whole Solentim team for partnering with EverCell Bio on this and allowing me to speak today about the VIPS and induced pluripotent stem cells. So with that, I'd like to turn the discussion to VIPS and induced pluripotent stem cells, various challenges, and what we feel a need for a new workflow. So for those of us who've been working with these cells, it's really no surprise that iPSCs are incredibly sensitive. Due to their inherent traits, they really require clump passaging in most cases and really rich and robust culture systems. Critically, they do not culture single cells without the aid of ROCK inhibition. And a number of studies have shown that repeated single cell culture of iPSCs can ultimately lead to genomic instability. And this really has greatly increased the challenges of cell culture and various applications that we're attempting to use induced pluripotent stem cells for. Now, some of these applications that require single cell manipulation can be shown here, and various applications such as gene editing, reprogramming, cell line rescue, or even clonal variability studies. And in large part, they're still performed very manual. It's very time consuming. And one could really argue that we've been somewhat unable to study clonal type of studies with induced pluripotent stem cells as single cells. At the very least, there's large opportunity to improve both the understanding of the biology as well as the process itself. So here at EverCell, why are we interested? EverCell is a single cell solution for induced pluripotent stem cell services. We can assist groups with cell modeling work, such as reprogramming, gene editing, or differentiation, or we assist with various product and instrument developments, focusing on QC, independent validation, or even just analytical support. So ultimately, EverCell is very iPSC centric, meaning that we really focus on induced pluripotent stem cell biology and technology and focus outwards versus the other way around. And really, given the utility and the complexity and challenges with induced pluripotent stem cells, we feel that we can accommodate these cells. We're very confident in being able to apply these techniques and workflows more broadly to other situations. And really, EverCell is here to provide very customizable and efficient services. And ultimately, we're looking for various traits in anything that we onboard or technology that we look at. And so given that the field is relatively young and making advances every day, we feel that there really remains large opportunity to further enhance various portions of induced pluripotent stem cell workflows. And some of these areas really involve, we feel there could be increased standardization among iPSC technology. We're looking for compatibility as this field is still rapidly evolving. It's important to both maintain instrumentation workflows that can both adapt now, but that are also forward thinking and leaning. And finally, if we can reduce timelines, it would be critical. And generally speaking, as a colleague once told me, the work feels very relatively qualitative. It kind of feels very warm and fuzzy. So we're very excited about any technology can make the process and or the biology more quantitative, repeatable, and supported. So when looking at the VIPS and induced pluripotent stem cells, as alluded to by Ian, really the technical hurdle was this. The VIPS instrumentation had required a dry plate or an uncoated plate for good droplet formation, whereas induced pluripotent stem cells inherently or traditionally required pre-coated matrigel plates or coated plates. And so this was really the first hurdle that was overcome. And this was something that we really quickly wanted to validate here at EverCell Bio before then further testing at other assessments. So the assessments that we did look at beyond validating MatriClone and VIPS seeding system was looking at the actual accuracy of the seeding and the efficiency of the outgrowth. How amenable was that? And was the system going to be compatible with various medias and culture systems? As just alluded to by the poll question, there's many different ways to culture these cells that hasn't really been resolved at this point. So it's important for instrumentation that we onboard to be able to be compatible with various different systems. And then obviously we want to look at the quality of the subclones and the biology. And lastly, how well does this work in our current workflow? And does it actually help us reduce our timelines? So first, I'll go through the setup and some of our pilot studies that we used to make some of these assessments. And for the pilot studies, these are the main areas of focus that I'll discuss here in the following slides. First, we examined droplet size adjustments. I also then want to talk about the detection of the single cells, as I feel it's one of the most powerful components of the system. We'll then talk about cell concentration and adjusting that and how that may impact seeding efficiency. And finally, we'll look at the outgrowth and timeline. We certainly want to understand how long this takes and how this can compare to current methods. And then again, do these clones maintain pluripotency marker expression, and how well can we actually expand these cells for downstream use? And as alluded to by the schematic below, this is the general workflow that we're going to be discussing for these following series of experiments. Ultimately, cells were passaged typically, traditionally, as clump passages on coated plates with MatriClone. They're then single cell dissociated and counted and placed in the reservoir, in which case when the VIPS, it's single cell seeded into a 96-well plate. We then monitored the outgrowth and then wanted to understand selection process and passaging out of the seeded plates. So the first parameter that we want to examine is really just the droplet itself. Ian gave a nice introduction to the droplets, but I wanted to show what they actually look like in our view. And by default, the cells are being dispensed through a nozzle. It's approximately 200 micrometers in size, and a volume is approximately 20 to 30 nanoliters. So looking at the far left at 0%, this is a default size of the droplet, and you can see that you get very nice clean border of the droplet itself, and it's also in the center of the well without any splashes. All of this is very critical to ensure that we're getting accurate detection of a single cell or not. And just looking at duplicates above and below, you can see that the droplet formation is very repeatable. Now, it's really nice that you can increase this droplet size as shown by plus 20% and plus 40% for a few reasons. One of which being the different media's components that possibly are used with induced pluripotent stem cells. And rather than focusing on the biology of these medias, it's more about the composition of these medias. For example, certain rich medias may require larger droplets, whereas cleaner medias, or perhaps have less serum in them, would require smaller droplets in order to get your optimal balance of cells and volume of media dispensed within that droplet. So we found this very nice, nicely and easily able to be adapted within the setup of the workflow upon seeding your initial plates. Keeping in mind that the instrument during seeding can be set to up to 4 droplet attempts per area and up to 4 areas within a 96-well plate. So next, beyond understanding the droplet, I want to highlight the detection of this instrument as it's very powerful and really clean. And so if we look at the schematics, typically a well is indicated by a green circle as indicated on top, or it's overseeded with a red circle and an X across it. Focusing on the top left first, you can see a nice droplet formation, of which we've zoomed in a certain area, which you can very easily do within the VIPS instrumentation in real time. And you can see that it very clearly identified the single cell. You can also see some debris next to the cell and or bubbles, and often this has been troublesome for certain AI and detection methods to find. But you can see very nicely, even the bottom left panel, that even when debris or bubble is on top of a cell, it very nicely can identify the single cell and only the single cell versus debris and other things around it. And this goes to what Ian alluded to earlier, is the powerfulness of their algorithm and their artificial intelligence in designing their detection methods. Conversely, if you look on the right side of the graph of the slide, you can see that these situations where there was overseeded. And in both situations where perhaps there is more than one cell seeded in a well, you can see that it quickly identifies both the cells beyond any of the other debris. And then on the bottom, you can also remove that, let's say it's in the middle of the division, or there's doublets, or there's just not quite fully dissociated cell types. It can quickly identify that there's clumps of cells rather than a true single cell, and show and indicate that that well has been overseeded and move on. So we felt this was incredibly powerful and certainly gives you a level of assurance as you head into the clonal outgrowth. So beyond the detection and the droplet formation, the really other parameter that we felt was most key to look on was actually the cell concentration that you're loading into the cell reservoir. And here I just want to highlight a few things. when we compared a low density versus high density. So again, green will indicate a single cell, red will indicate overseeded, and gray well will indicate an empty well that's moved on. And looking at the top, this situation, we loaded cells at 5,000 cells per ml, and then proceeded with the seeding procedure. And you can see by the summary graph or pie chart on the right that overall we did still get nice single cell efficiency, but we did get a large amount of empty wells, as shown about 11%. Now, conversely, if we, on the bottom half, if we used and inputted a cell concentration of approximately 15,000 or higher, we certainly eliminated any empty wells, but you can see that we have a higher propensity for overseeding, in which case we can assume that we can dial this back down and really find that happy balance. And something I really want to note here too is this is a balance of time and droplet size and concentration of cells, and it does impact the timing of what it takes to actually seed a whole plate. So with a lot of empty wells, it's proceeding through the whole seeding 16 attempts and can sometimes take a little bit longer, approximately 12 minutes or more, versus the high concentration or seeding with more cells than you might ideally want will accelerate the seeding process and can take less than 8 minutes. So it's a little bit story of Goldilocks here and trying to find out what is just right situation. And here's what we've landed on. And we input 10,000 cells per ml into the cell reservoir. We finds that really nice high efficiency with having a low propensity for empty wells and still maintain a nice high efficiency as high as 80%. And I just want to reiterate some of the examples below of various droplets being formed and the detection of the cell and the instrument being able to easily identify such. And it's important to note that through all this optimization, we really feel that cell concentration is more or less set, and really all you're playing with is dependent on media. So even though I have gone through certain optimization procedures, I want to note that it's very seamless and very straightforward to do so during a run that you're about to perform. Now, lastly, before moving on to some more biological questions, I just want to touch on the different medias and being able to adapt the instrument based on droplet size to accommodate various medias. So here we tested cells that have been cultured and loaded with two different types of medias. Media A being FGF stabilized and rich in serum. Media B being much less rich with serum and a little bit cleaner by definition. And in the top media with a little bit larger droplet size, we're able to find nice seeding efficiencies. And for this, we seeded a total of 5 plates per condition and tried to get a nice average and understand the robustness and the consistency of seeding those plates. And on the top media, we actually had to adjust for a little bit larger droplet size. Looking at media B, we also were able to adjust it and get very nice seeding efficiencies comparable to the other media. And this was simply by adjusting the droplet size just a little bit smaller, and it was still able to maintain that droplet and have good detection parameters. So with all this in mind, we took care of the 2-in-1 system, talking a lot about the seeding parameters. The other set of capacity that the instrument VIPS has is the whole well imaging, which I want to touch on a little bit as well. And really here, I just want to have a schematic on the top, understanding the timelines of when we imaged. And so following seeding of the plate, we would bring the 96 well plate back for imaging 2 hours post-seeding, and then follow it for the first few days to try to understand the very initial divisions and understanding that the clonality is true. And then we followed imaging throughout the remaining outgrowth from day 5 to day 12 to better get a sense of the timing of outgrowth and when is best for selection and when is best for expansion. Now, for each time that you perform whole well imaging, you do get a nice heat map on the lower left that you can see. You can also see that hasn't been touched on well either yet is that A1 is actually a control purge well. So when we're talking about seeding, it's out of 95 wells, not 96, and also shows as a good indicator of how the whole run went, often with monolayer of cells indicating that it was properly run and you have nice outgrowth. And then on the bottom right, you see an example on the left image of the whole well, which you can then very easily zoom in on the instrument and highlight and locate that induced pluripotent stem cell colony. And as you can see here, you can see a nice induced pluripotent stem cell colony, even if it's up close to the ledge. To drive that point home a little bit more, another image of the whole well on the top left, and that same image of the iPSC colony shown on the top left. There's some representative images of induced pluripotent stem cell clones that have been that had grown out from the initial seeding event on the top right. And I really want to highlight more the fact that, one, on the instrument itself, you can zoom in and out and really quickly identify a rare population within a whole well very quickly. And two, even if the well is against the edge of, even if the cells in the colony are growing against the edge of the well, you can very quickly see that, and the instrument can very readily pick up these cells. And it's not shown here, but I do want to mention that there's confluence detection on the VIPS and can very easily identify these colonies, both from a heat map and as you zoom in. And we're very excited to continue to using this confluence detection to further enhance the days and outgrowth and timing of which we select various colonies. So taking all of the optimizations into account, we really want to think of a situation that we might apply this to. And so under the assumptions for gene editing project, we might want to look at least 100 clones. We went ahead and decided to run, under certain conditions, 3 plates and see how consistent and robust not only the seeding was, but the outgrowth. So here are an example of 3 plates and the schematic of the seeding report on the left. We then allowed them to grow for 10 to 12 days. And at 12 days, ended the experiment and decided to manually confirm various wells that had clones, iPS stem cell clones, grow out from the initial seeding events. And what we observed after all the optimization on this particular run was very nice, high, robust seeding efficiencies and outgrowth efficiencies as well, as high as 60% and very robust and similar with 57 and 58% respectively. And we're very pleased to see that this really alluded to almost 46 clones, nearly half the plate, being true verified clones that can be outgrowth and selected for downstream use. And compared to current manual dilution methods, it certainly adds a level of validity to the biology that we're later taking on and spending a good amount of research and time and money and effort on. So now that we've understood that we can seed these plates, before we even do any selection or expansion, we wanted to confirm that pluripotency is maintained in the well. So we took an example well and decided to fix it at day 12 and perform immunocytochemistry with HRP using NANOG as our marker, and very nicely highlights the whole plate. And you can see the various clones that have outgrown from the initial seeding events. And then on the right, you can see an expanded close-up view of a particular clone in one of these wells. And you can see nice morphology, nice NANOG staining throughout the whole colony. So we're very pleased with this, and certainly we're excited that the cells in the seeding, VIP seeding events itself has no impact on the pluripotency nature of these stem cells. Next, we want to understand how easily can we actually expand these out of the well. This is typically overlooked, but also very troublesome and tricky in some cases for current workflows to actually just manipulate these cells and get them to the expanded state that you need to while still performing the screening and other applications that you might need. So for one example, we took a plate and decided to expand at least 18 clones from that plate. And in doing so, we just reverted back to clump passaging and was very easily able to expand a single clone into a 6-well plate first for further expansion and downstream assessment. So very nice to see these outcomes thus far, and excited enough that rather than set a whole other set of experiments, we wanted to go ahead and take one of these clones and perform even further characterization on it. So after a further expansion, you can see a representative image on the top left. We then performed further pluripotency marker expression with Trawin60 and NANOG, and you can see that it's stained positive for those markers as well. And alluded to earlier in the talk that single cell manipulation of these cells can often lead to genomic stability problems with induced pluripotent stem cells. So we definitely wanted to assess the karyotype of this clone. And we were very pleased to see the results coming back as complete normal karyotype. Following karyotype, we performed differentiation using EverCell's differentiation platform into mixed cortical neurons. And you can see we achieved very nice differentiation with markers of beta tubulin and MAP2 indicating our mixed cortical population. So with all this in mind, we are very excited, and it worked with one of our lead course or workhorse lines. But as most of us in the field know, there's high variability in lines coming from various sources. So we want to ensure that we could test this and validate it across multiple other cell lines. So for these set of experiments, Just to discuss the workflow, we adapted these iPS stem cell cultures to MatriClone and clone passage methods for at least 2 to 3 passages prior to seeding. We then seeded these cells in the media of choice, along with MatriClone and with ROCK inhibition, in all combined and complex together, and seeded those into 96-well plates. We then monitored the outgrowth via cell metric imaging and selected a number of clones for clump expansion, and finally selecting a subset of those clones for cryopreservation and further characterization as described prior. The lines we assessed consisted of 2 affected and 2 presumably healthy lines, and the characterization was that similar to performed earlier, which was NANOG trauma 60, ICCIF, karyotype, and neuronal differentiation. And you can see a schematic below, a little bit more expanded from earlier, in which we're trying to capture and understand the timeline of full from seeding all the way through selection, expansion, and then downstream characterization. So it'll be fairly high-level review, but first I give a schematic of the overall seeding events as well as the outgrowth. Again, you can see 2 healthy donor iPSC lines and 2 disease iPSC lines. You see the seeding report graph on the far left, an example of a droplet formation from each one of those events. We then monitored the outgrowth for approximately 12 days and then performed expansion into a 6-well plate for cryopreservation as soon as P1. Upon thaw and cryopreservation recovery, we continue to expand and then perform pluripotency marker expression assessment. And here again, we see for a total of 3 clones over 4 lines, we see that pluripotency is maintained in all clones that have been selected. We then took a subset clone of each one of those donor lines and sent these off for karyotyping. And again, very pleased to find that we had healthy karyotypes and not concerned about some of the instability coming through the VIPS seeding process. And finally, just to confirm their potency and ability to differentiate, we again took each one of these subclones and ran them through EverCell's differentiation platform to make mixed cortical neurons, as shown here by beta tubulin and MAP2 positive stains. So we were excited with all of this data, and really more excited about how easy it was to onboard this technology and to get induced pluripotent stem cells to use. For a lot of other technology, it seems to require a little bit extra heavy lift. So it was really nice to see that some of our earlier considerations were very easily squelched using MatriClone. And by using MatriClone and complexing it with the media as well as ROCK inhibition, we're really able to get iPSCs to be plated on plates that have not been coated yet. And that's really not the traditional way in which they're currently cultured. And we really feel that we checkboxed all the assessments that we were looking to do with these initial pilot studies. We certainly were able to see the induced pluripotent stem cells using the VIPS and the MatriClone complex. We saw very nice, accurate seeding and very nice high efficiency of outgrowth of these cells. We were able to show that the VIPS is compatible with multiple different culture systems, which ultimately will just make the adoption of this technology a little bit more seamless. And the quality of the subclones were good and passed all criteria that we typically put our clones through. And finally, we feel that it was, again, very easy to adopt within our workflow, and we feel that there's a huge potential to reduce the timelines. And if we look at the timelines, this is really what we settled on as our final workflow. And what's impressive is really that before day 12, we can have single clones available and really be screening them before day 16. And if we carry it all, put it all together in a streamlined workflow, we see that by day 20, we can have clones expanded and banked and early screened for something that we may want to apply for later on, before then having to use them in our downstream applications. And this certainly, compared to traditional methods, is at least twice as fast, I would argue. And all the while in the current methods, there's really no validation of clonality. And so we're a little less confident in the biology for downstream uses of them. Taking everything into account, we seeded at least 60 plates and overall saw seeding efficiencies around 72%. And of the seeding efficiencies, we saw outgrowth in the range of 40–60%. Or 27 to 45 clones per plate. And for us, this was certainly met our initial goal, which is to see at least 25 clones per plate would enhance our certain workflows, particularly for gene editing. So for our next steps, as I alluded to, we're very interested in applying the VIPS system to our gene editing workflows. And I should mention that we do utilize other stem cell types and adult stem cells. So we're also interested in applying the VIPS system and clonality to other types of cell types, whether it's iPS derived or adult stem cells such as MSCs. So finally, before I turn it over to Ian for the conclusion of the slide presentations, I want to touch a little bit further on some of the iPSC applications and where we foresee incorporating the VIPS into some of our workflows. And just very briefly, looking at a couple typical induced pluripotent stem cell application timelines on the left, subcloning, reprogramming, and gene editing. Really, this is the typical timelines that we have to handle or deal with when working with these cells. And I think it's important to note that even with these long timelines, we're still doing this all very manual, and there's high variability within the process. In current state, EverCell for gene editing manually limited dilution plates induced pluripotent stem cells post delivery of the constructs, and then has to pick these clones manually into a format that can be further expanded. And then from that point, we can screen these certain colonies and hope that we can get the screening process done in time before having to put too many resources into the expansion of the various clones. Now, if we have a VIPS system, we foresee being able to expand the induced pluripotent stem cells to the state of needed, delivering the constructs, and immediately using the VIPS to seed these cells and monitor various outgrowth. This would allow us to then potentially screen these cells prior to passage 1, and certainly before passage 2, identify the cell clones of interest before then proceeding with banking, characterization, and downstream differentiation or applications that you might use these cells with. Furthermore, I think it could be incredibly helpful in this situation that currently we have no validation that the cells or the clones, colonies that we're picking come from an actual validated clone. So the events, we could be picking colonies that have arisen from 2 different clones, leading to mixed populations, and ultimately leading us to have to re-subclone an entire population again. So we're envisioning that the VIPS will help remove that potential obstacle and help streamline and reduce the amount of times that we potentially have to subclone mixed populations of our gene editing events as well. And we foresee using the VIPS to be able to really reduce timelines for all of these workflows. And again, even more importantly than reducing the time, adding that level of validity to the biology and confidence that we can use the clone that we really want that's precious for the downstream applications of interest. And so finally, whereas current state is still fairly laborsome, very manual, and certainly not validated, we foresee the future state of a lot of iPS stem cell workflows, such as gene editing, reprogramming, and beyond, including incorporating the VIPS system to be simple, robust, and highly validated. And we certainly look forward to incorporating this in further applications. And with that, I'll turn it over to the next poll.
Thank you, Phil. So that does bring us to our final poll question, audience members, and you should be seeing that poll question on your screens. And that question is, what media do you currently use for your iPSCs? And your options are mTeaser Plus, StemFlex, Essential 8, NutriStem, or PeproTech HESC. So you can go right ahead and click on the option that suits you best. And once again, that question is, what media do you currently use for your iPSCs? So your options are mTeaser Plus, StemFlex, Essential 8, NutriStem, or PeproTech HESC. And also a quick reminder that you can still send in questions for Ian and Phil. We are getting lots of interesting questions, so go right ahead and send in more questions using that questions module just there. All right, so it seems like the majority of audience members have voted, so I'll go ahead and close the poll and share those results with you. So you can see the results right now. Okay, so it seems like the majority of votes went to mTeaser Plus at 39%, followed by Essential 8 at 23%, and then it was StemFlex, followed by NutriStem, and then PeproTech HESC. All right, thank you so much for participating, audience members. Now back to you, Ian. So Ian, I'll send controls to you right now.
Okay, so I just want to, can I just confirm you can see my slide, support for regulatory?
No, Ian, so we still don't see your slides. Do you want me to take back control, send that to you again?
Yeah, I didn't get a control message from you.
Okay, I'm doing that again.
Okay.
All right,
Okay.
that looks great.
Perfect. Thank you very much for your assistance. So thank you to Phil for the data element of the presentation. I just wanted to sort of take you now through the steps of how we plan to support particularly customers developing stem cell therapeutics and for regulatory and clinical aspects of this. So I want to start with this. I do sort of make a bit of a disclaimer here. These are not stem cells, but I just want to illustrate the point of how you would generate and utilize this information to support documentation of clonality. So again, I'm just going to bring a laser pointer up so I can illustrate what I'm talking about. So we've got a time course here where this first image on the left is that droplet image we saw before of the VIPS, and this is on day 0. And the AI software has detected, or the Z-Stack software, analysis software has identified the single cell highlighted there. And then we've got daily whole well imaging then. And so this is when the well is being filled up with MatriClone or media, and we're confirming the presence of a single cell in the whole well environment. And we're looking at various time courses. So here they've actually looked at it a couple of times on day 0. And then they've looked at it 24 hours later on day 1. And this is where you see 1 cell becomes 2, it's divided. And then down here, 2 cells have become 4, and you can see a colony. Now, all of this data is captured every time you put the plate into the VIPS or the cell metric to capture these whole well images, and this is your time course. So I think some of you will maybe thinking or wondering, well, is documentation of clonality automatic? And I'd have to say it's not, for a reason. I don't think you'd ever want it to be, and you'd probably never trust it if that were the case. But we saw the green and red colours on the plate map earlier, denoting a green one was where there was a single cell. Now, what that means is it's highly indicative there's a single cell there based on the AI and the detection of the cell in the droplet. But you'd still want to inspect that before you committed it to a master cell bank, etc. So I think in terms of practical workflow, you'd be perfectly acceptable to take forward all the green wells, for instance, from the plate and expand those. But I think you'd want to come back at a later point before you bank it and say, okay, well, let me just go back to the day 0 images in the well and double check I've only got 1 cell there and absolutely nothing else in the well. So it's still prudent to go back and do the extra checks of those really high value clones you want to bank. The other point I just wanted to make as well is the beauty of doing this imaging in the droplet and day 0 is it doesn't really matter then if the growing colony or the cells move later on, because you've already captured and documented that it was a single cell at the inception. So some people get a little bit worried about colonies that move or split. Well, really, when you've got this very early strong information, you don't need to worry about that. So the actual report then, I've said in the sort of title here, you literally would include this as part of your IND. The VIPS will generate this type of report, and I'll just explain the sets of the report. So you can decide what ends up in the final report. You don't have to put every single time point in. You want to tell a story to the regulator. So you've got a time course here at the top where my mouse is at the moment, my laser pointer, and these all represent different time points or different days. So you certainly want time 0, and then you can choose subsequent days. We're seeing here we've taken one at day 2, another one at day 2. You might want one at day 7. And then the aspects of this report are, first of all, we're looking at everything in the whole well scenario. We're not just taking a snapshot part of the well in isolation. We're looking at the whole well, and it's important the regulator can see this. But then we're taking the region of interest where the single cell is, and we're exploding it here. So we can see that's nice and clean, and that's a single cell. But we can also then pick out other regions of the cell where there might be little blobs or marks or facets that you want to highlight for the regulator, and then explain why these are indeed nothing to worry about and they're nothing to do with the cell and don't impact clonality. And then when you go to your second time point, your second day, you're automatically picking up this same region of interest again, and there's now 2 cells, which is great. Regulator would be pleased with that. And you can see, you know, again, you can report on what happened to those other facets as well. All right, so now I just wanted to talk about how Solentim plan to support clinical groups moving forward. So I think we appreciate first and foremost that customers buying instrumentation to do stem cell work and potentially stem cell therapeutics need to be making decisions about buying equipment that's going to go into research that can then also be replicated in their manufacturing and GMP groups. They don't want to have to start again when they get into GMP. So we've really sort of laid out our case here by adopting something called GAMP5 and policies for that. So Solentim has worked to put GAMP5 in policy in place, and this is recognized in the industry by procurement groups and quality groups for computerized validation systems and GMP groups. And we believe this is critical for starting to work with customers who are developing therapeutics. And I think, you know, there's going to be lots of specs and differences, I suspect, across equipment that can work in this area. But I think the single most important prerequisite when deciding which equipment companies need to purchase to support them will be whether they can actually be used in a future clinical manufacturing environment. So I'm sure this terminology will resonate with some of you, and you'll be pleased to see that Solentim have taken this stance. Also, you know, we've launched a new consumable today, MatriClone, Freudian slip, MatriClone, and that's currently sold by Solentim for research use only, and that's available on our website. New web pages have gone up today, so you can get information on that and potentially order that. But importantly, for those of you in the clinical areas, you'll be thinking, well, what about, you know, when I get to GMP? And just for clarity, I want to let you know that there is a GMP grade of the matrix available, and we can help you source this. But at the moment, this would need to be a discussion with individual customers and would be on a request-only basis. And because of the nature of the manufacturing and manufacturing to forecasts, it's a longer lead time to get this, but it's all possible. And hopefully that, again, will give comfort to some of the people listening that are interested in evaluating MatriClone. So finally, I'm conscious of time, that we want to allow 5 minutes or so for questions at the end. So next steps, well, as I say, MatriClone is now available from Solentim. It's on the website, it can be ordered, and it's being stocked in the US and Europe for immediate purchase and testing. And Solentim are geared up for now taking this the next step with you, the listeners, to do stem cell demos on the VIPS. These, in the first instance, can be a virtual demo, taking you through the software steps and some of the imaging and how we set up and use the machine. Or it can be, with a bit more planning, an on-site demo. And if indeed we're allowed under the current sort of COVID situation, in some instances, we can then come to your site. I would just point out that you need to purchase some MatriClone before we come and adapt those cells in advance of us coming and doing the seeding. And, you know, you've met Phil verbally, vocally today. He's based just outside Boston. So customers in the sort of Boston, Massachusetts area are very welcome to go and see Phil, talk to Phil, and see the VIPS in his lab. So with that, I will just quickly do some acknowledgements. Thank Phil and his team in EverCell. And at the Solentim end, Claire is our head of biology. Joey is a local applications specialist. He's previously worked in industry and is now based in New Hampshire. George Bishop is one of our heads of software, and George has helped with all the AI software for iPSCs and the confluence detection, and myself there at the end. And with that, I would like to thank you very much for listening and turn back to the organizer for sharing some questions. Thank you.
Fantastic. Thank you very much for that very insightful and detailed presentation. So that does bring us to our Q&A portion of this webinar. So audience members, you can still continue sending in questions for Ian and Phil. We've received a lot of questions already, so let's go ahead and start off with the first one. So Ian, this question is for you. Do you see much better viability of clones when using the VIPS plus MatriClone combination?
Yes, we do. I tried to sort of point that out, that I think we on the first hand we see better performance of MatriClone against probably one of the more incumbent matrices like Matrigel. But then when you combine MatriClone with the VIPS, you get even further uplift compared to limiting dilution. So, you know, I mentioned sort of 4 to 5-fold more colonies probably than limiting dilutions. And I think Phil spoke to this and the number of colonies he was getting in a 96 well plate.
Great, thank you, Ian. And another question for you. And that question is, if I already use a cell metric imager, can I still use MatriClone?
Absolutely. I would encourage cell metric users who are working with stem cells to purchase a MatriClone and try it. And it can be used in the soluble form that we've used it, which is, so you can do a low concentration, or you can use it in the coated form. And they should see better, you know, image quality probably than they've seen with vitronectin.
Fantastic. And another really interesting question from an audience member. And that question is, how much lab space does the VIPS take up? And does it need to be in a hood?
Yeah, we're going a biosafety cabinet, just a standard one, because when the plates are inside the machine, the lids are off during dispensing, so it's important to keep it clean and sterile.
Interesting. And another question, and this one is for Phil. So that question is the VIPS system compatible with enrichment strategies like FACs?
Great question. Yes, we certainly feel like it is. You know, our model, generally speaking, is the least manipulation, the better, but certainly in situations for FACS or enrichment, you can perform a MAX or a FACS prior to loading those cells into the VIPS. So we very much see it as complementary, and the VIPS still adds that clonality factor and the confidence in the cell that you're still selecting.
Great, thank you, Phil. And another question for you. So that question is, what is the most critical part of the workflow to optimize?
That's another great question. I think it's somewhat of a trick question. I think for the VIPS instrumentation on optimization there, it's really just a balance between the droplet size and the amount of temps that you're trying to make per plate. And a lot of that is more just based on the needs of time and how many cells you need. So you really don't need any optimization if you just need a couple clones out of a whole plate. It's more about if you really need to squeeze the most out of a certain application for what you're doing, where you might want to optimize those two particular cases. The last part that's important to optimize, which is really just more about induced pluripotent stem cell biology, is as with all the other applications, it's really dependent on the quality of the cells that you're using and loading into the VIPS system itself.
Thank you, Phil. And we have time for one more question. So that question is, was there differentiation observed in seeded clones?
No, there wasn't, at least not in terms of if they were healthy cultures going into the VIPS. We've got very nice clones out of the VIPS. And in fact, I think the conditions actually somewhat select for the more pluripotency form phenotypes that we see.
Fantastic. Well, Ian and Phil, thank you very much for all of those answers, and to everyone for sending in all those questions. Unfortunately, we have reached the end of the Q&A portion of this webinar, but not to worry, if you couldn't attend to your questions, the team at Solentim may follow up with you later on as well. And if you have further questions, you can direct them to the email addresses. So let me go ahead and show that to you. So go ahead, quickly jot those email addresses down. So once again, thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. So a survey window will be popping up on your screens. Your participation is appreciated as it will help us improve our future webinars. Now, I'm about to send you a link in your chat box. You'll be able to view the recording of this event at that link and also share this link with your colleagues when they register for the recording as well. Now, please join us in thanking our speakers, Phil Manos and Dr. Ian Taylor, for that fantastic and insightful presentation. On behalf of the team at X Talks, we thank you for joining us. Take care. Bye for now.